for fertilization. All right, well, let's see what happens during fertilization. Okay, a number of things happen somewhat before fertilization occurs. Now, once again, you think of kind of like the, you want to call it the evolutionary pressures that are going to lead to things like various aspects of fertilization. One of the most important things is your sperm have to get to the target very, very quickly, faster than anybody else's. If you're an organism, and let's face it, most organisms on this planet do external aquatic fertilization. So what that usually means is your sperm are going to be competing with the sperm of a whole bunch of other males. For instance, case in point, there are sea urchins. If you keep sea urchins in a tank at too high of a density, what will happen is if one male shoots out a little bit of sperm, the other males pick that up and they all shoot everything they got out. So it's like, well, if somebody's having sex, we're all going to join. It's kind of like what you see in a fraternity party, right? Let's face it, there have been some fraternities that have been kicked off of campus for that. Usually involving drunk or otherwise unwilling women. Oh no, there's people gone in jail since I've been here for some stuff like that for some of the fraternities. But anyway, what's going to happen is if there's some eggs around, you know, every male in sight is going to be releasing their gametes. So everybody's rushing towards those eggs. And the winners are going to be ones that have the greatest sperm volumes and the fastest sperm. Okay, so that's one kind of thing. Now, organisms with internal fertilization may also have to face certain problems too, because sometimes you don't really know if your lady is actually going to, when you're done with her, she's going to go and find a better guy and he's going to do it. So, you know, there are a number of different tricks where males compete with each other. For instance, fruit flies. Be thankful you're not a fruit fly female. Because what the male fruit flies do is when they fertilize the female, they put in a substance that solidifies in the reproductive tract and essentially forms a chemical chastity belt. Not only that, and that is going to make it difficult for another male to fertilize the same female. Not only that, some of the substances in the semen of the fruit fly are somewhat toxic, so they make the female sick. So even if another male does come by, it's like, oh, not tonight, honey, I feel terrible, I got a bad headache. Well, yes, because she's got poisoned by her previous boyfriend. Sounds like you can see articles in newspapers like that here. But at any rate, you know, there's a number of different tricks for one male to prevent fertilization of the same female by other males. And even at the level of sperm, that's what's sometimes called sperm competition. Now it hasn't been really rigorously shown yet, but there's a suspicion that some sperm in a population of sperm from one male are going to act more like linebackers in a football team that will block other male sperm, while other populations of sperm are actually rushing towards the egg. So it's like, okay, in a football game, only one person has the football, right? And he's running towards the goalpost, the egg. Okay, he's running towards the egg, and what does the rest of the team do? They're trying to block the people that are trying to tackle him. That apparently happens. It's widely suspected that happens with certain sperm. Some sperm, rather than rushing to fertilize, are actually going to try to block competing sperm from fertilizing. Mechanisms are still kind of unknown. It's possible they may secrete things. It's possible they may fire off their acrosomes, which are full of nasty enzymes at other sperm, and things like that. But you often have that, especially if you have sperm from multiple males inside the same female. So there's all kinds of little interesting tricks that males do to make sure that their sperm gets to the eggs and somebody else's doesn't. Not all those tricks are very nice either. So that's one kind of it. Now, second thing here. Since sperm are designed to be lean, mean fertilizing machines, what we've done, we've got the maximum propulsion possible. We've shedded as much weight and as much flab as possible. We've made the sperm as compact as we possibly can so it moves faster than anybody else's sperm. Okay, the problem is that flagella burns ATP at a tremendous rate. The sperm mitochondria can only produce a limited amount of ATP before they run out, and when they run out, the flagella stops. So what we need is to make sure that the sperm flagella is inactive until you know you're going to need it. And that's what we call the process of capacitation. Capacitation, the activation of sperm motility. In other words, allowing the flagella to work. Okay, now how do we do that? It depends on the organism. Many times organisms that are going to fertilize in aquatic environments will do something where going into the water is going to activate the sperm motility. For instance, when we look at sea urchins, the sea urchin's coelom is loaded up with substances that will actually block sperm motility, inhibitors of sperm motility. When you release the sperm into the seawater, those substances are diluted down. Not only that, inside the sea urchin, the pH is about 6.8, just a little bit less than neutral. Sperm motility is inactive at those low pHs, at least for this type of sperm. But now, you release it in the seawater, you're diluting down the motility inhibitors that were basically in the semen, and the pH goes up to about 8 or 8.2. And lo and behold, what happens? Sperm motility is activated. Now, as we saw in the lab for sea urchins, starfish, and stuff like that, those sperm can last about 10 minutes before they run out of ATP. And we've seen many times, you looked at the slides, and there's a whole bunch of dead sperm lying around, or at least inactive sperm. So, you want to make sure that you're close to a pile of eggs before you release them. Now, here again, in other organisms with aquatic fertilization, it may be things like changes in salt concentration, like organisms that fertilize in freshwater, dilution of inhibiting substances, things like that. Okay, now what about organisms that practice internal fertilization, which basically includes most tetrapods, especially amniots. Okay, well, what happens here? Male has a structure of some kind that's inserted into the female's reproductive tract, called by a whole bunch of different names. Some of them we can't repeat in class, but there are also technical names for the male reproductive organs and things like reptiles and things like that. What's going to happen here? Sperm gets squirted into the female's reproductive tract, whether it's a vagina, whether it happens to be a cloaca, whatever the case is. And once again, if you squirt that sperm powerfully enough, you can go deep into the tract, and that's going to help things out. You'll have a higher population of sperm that are going to be able to make it. The further down you can shoot the stuff off. So you already help save some of the ATP by putting the sperm fairly far in the reproductive tract. Okay, so you can do that. Now, what's going to activate those sperm? Well, capacitation in organisms with internal fertilization is usually something that is in the reproductive tract wall. In the case of mammals, there are lipids in the vaginal and uterine walls that activate the sperm motility. Okay, so in our cases, we've had, and this includes humans, lipids in the female reproductive tract are going to activate motility. Now, I'm not sure on this, but I believe in vitro fertilization they have to put some extract that contains those lipids when they do it in a petri dish and things like that to activate the sperm motility so that fertilization occurs. Okay, so that's going to happen. And that ensures that the sperm are not going to be activated until they're actually in a location where they're likely to have a shot of fertilization. Okay, so capacitation is one thing because once again, sperm have limited supplies of ATP. Now, by the way, human sperm can actually still be motile for as much as about 3 days afterwards. Okay, so anybody that uses things like calendar rhythm methods or whatever for contraception, you have to realize those sperm could be active for 3 days, possibly even a little bit more. There's a debate about that and stuff. So it's still possible to get pregnant later than you might expect. Okay, so at any rate, that's capacitation. Your sea urchin, you have about 10 minutes. Okay, so that's one kind of thing, the capacitation, the activation of sperm motility. Now, second thing is this, and this is true for aquatic or external or internal fertilization. If you're a sperm cell, let's say a mammalian sperm cell, your sperm head is typically 5 to 10 micrometers in size. It's very, very small. It's one of the smallest cells in the body. Now, using a human female as an example, that trip they have to make up the fallopian tubes and stuff like that is 10 or 15 centimeters. That is an enormous distance. To go 1 centimeter, 5 micrometers, that's 1/1000th of a millimeter, so that's a 200th of a millimeter, a 2,000th of a centimeter. So, sperm's going to have to travel roughly about 40,000 times its length to be able to get to the egg in the fallopian tube. Now, on a human scale, consider, say, a 5-foot tall human, 40,000, 200,000. That is about 40 miles. So imagine the equivalent of a 40-mile walk for one of us. That's a lot of effort. That's a long distance. Now, you got a second problem. In this enormous reproductive tract, or this enormous amount of aquatic environment, you've got to find those eggs. After all, if you're going to do a 40-mile hike, you might want to get to the destination that you're looking for and not get lost somewhere on the way, right? I'm sure you have to experience that, right? Okay, so how do we find the egg? Well, it seems in most species, and there's evidence now that applies to humans as well, we can easily see it with the sea urchins. The egg helps the sperm along. And this is a process that is known as chemotaxis. Okay, what's going to happen? The eggs release molecules, usually small molecules. In fact, I think many of them are lipids. Some of them may be modified nucleotides, things like that. The eggs release small soluble molecules that are going to diffuse from the egg and then slowly dilute in the environment. So you have a chemical gradient. Closer to the egg, higher concentration. Further from the egg, lower concentration. The sperm cells have receptors for these things. The receptors will activate a signaling pathway that will alter the beat pattern of the flagella and basically cause the sperm to steer into the direction of the higher concentration of the stuff. So the egg basically says, oh, here I am, come and get me. Some of that Stony Brook a few times in parties. All right. Okay, but basically, the eggs are going to release these chemical attractants, and the sperm will alter the motility so they start heading in that direction. And very soon, you can get enormous amounts of sperm clustering around the eggs. Now, we saw that in the lab. When we looked in the lab, we could see very clearly enormous amounts of sperm clustering around each egg, and much less sperm in the surrounding media. That's our chemotaxis. That helps a great deal because the egg cells are often relatively small and they're often fairly dispersed. So you got to attract the sperm towards it. And we see that, and like I say, that seems to apply in invertebrates and invertebrates as well. Now, an interesting little speculation. Presumably, one could make some potential kind of contraceptive to suppose you had a little implantable thing at the base of the fallopian tubes that would release fairly large amounts of the sperm attractant, so that when something happened, the sperm would head towards this little thing that's releasing it and wouldn't be able to find the eggs. That might be a potential contraception. They can identify things, and a fairly specific one if people could identify exactly what the chemoattractants are in human sperm. So that would be an interesting subject for research. But anyway, or something that would block that receptor so it couldn't find its way, to the egg. That could be another potential contraceptive that would be specific as opposed to the hormonal ones, which have all kinds of potential side effects. Okay, well, anyway, we got something like that. So the eggs do help the sperm home in on it. Process of chemotaxis is also important in the events of fertilization. All right, questions on that, sort of like precursors to fertilization.